First, thank you so much to the organizers. This has been an incredible day already. I know that people are slowing down, so I'll try to keep this brief. But excited to tell you about some of the sort of a new direction we started thinking about. I switched the order of my title a little bit to focus on the effect of stress on species interactions. So mobile genetic elements like bacteriophage are ubiquitous. As I think we'll hear more tomorrow, the number of phage in the environment, the number of phages are astronomical. And just as one quick example of that, there are estimates that up to a third of the bacteria in the ocean are infected by phage every day. So I think what this suggests is that these infections and incorporation of the effects of these mobile genetic elements may be very critical to consider. As another example, plasmids, 43% of all bacterial genomes in NCBI carry at least one plasmid. So across a wide variety of different types of mobile genetic elements, the data suggests that these are really prevalent. And there have been some great ways, lots of theory developed for ways that we can think about the way that these mobile genetic elements are impacting microbial systems. Clearly, bacteriophage play a really important role in determining both the composition and the function of microbial communities, controlling the composition through processes such as kill the winner or piggyback the winner. And additionally, they're driving the viral shrunk, so leading to the release of nutrients, which plays a really central role in the global carbon cycle. Additionally, mobile genetic elements can play a really important role in allowing for the acquisition of novel function and novel traits within microbes. And we see this particularly in the spread, for example, of antibiotic resistance. We really frequently see novel antibiotic resistance enter new species and new cells through horizontal transmission based on plasmids. But I would argue that perhaps even more fundamentally, mobile genetic elements are likely to shape microbial systems because they fundamentally rely on the metabolism of the host. So most mobile genetic elements, such as bacteriophage and plasmids, don't carry their own replication machinery. They're obligate parasites. They rely on the metabolites generated by the host and much of the machinery generated by the host. And as a result of this rewiring of intracellular metabolism, that's also likely to drive changes in both the uptake and the secretion of metabolites by these infected cells or by these MGE-carrying cells. And this clearly has implications for then driving changes in the way that these infected cells interact with other species in the community. So we were interested in asking, to what extent do the metabolic rewiring driven by MGEs end up actually shaping microbial communities, species interactions, and emergent properties. To get at this, we use two sort of classic mobile genetic elements. So we focused on the F plasmid, the conjugative plasmid, as well as a filamentous bacteriophage M13. So the F plasmid is the plasmid itself. We used 128, it's rather large, and it encodes this expensive machinery which drives the formation of pili. And these pili allow the transmission of this F plasmid between cells. The filamentous bacteriophage is a type of bacteriophage, obviously, but one of the things that makes it stand apart is that it forms these chronic infections, these chronic active infections. And so the bacteriophage gets into the cell, produces phage particles. These particles then sort of extrude out of living cells. And so it's not, so it's active as opposed to, say, a prophage or a lysogen, but it is not bursting open a cell like a lytic phage. And so you can get these amazing pictures like this, where you can see many different viruses coming out of a single cell. Critically, I'll also point out that M13 enters the cell by attaching to the pilus, so of the F plasma. So that's another reason that we chose these two elements. And furthermore, an additional component of them being chronic is that they can be transmitted vertically as well as horizontally. We then tested the impact of these mobile genetic elements in a synthetic community that we've developed and worked with for some time. The community involves a methionine oxytroph of E. coli paired with a strain of Salmonella, which secretes methionine. And then we pair this with Methylobacterium extorquens. And when we provide lactose as the carbon source and methylamine as the nitrogen source, these three strains form an obligate cross-feeding mutualism. Conveniently, by simply adding either adding nitrogen or methionine, we can also grow all pairwise combinations of these strains. We then infected our E. coli. We used E. coli as our focal organism and infected it either with just the F plasmid or the F plasmid and M13, and simply asked, how will this impact the community? We started by trying to do some modeling and basically copied a Sawyer paper where we used their methods to account for the building blocks of viruses and incorporate them into genome scale metabolic models of E. coli. Genome scale metabolic models were touched on earlier. Basically, this is a model which incorporates all of the known metabolic enzymes and metabolic reactions inside of an E. coli. So we took a curated genome scale metabolic model of E. coli, which incorporates the methionine oxytroph mutation, which we know is in our strain. We then accounted for the nucleotides, amino acids, and ATP costs associated with generating each of these compounds and build a hybrid model, which now has three biomass equations, one just for the host cell, one for the plasmid, and one for the filamentous phage. And again, the protein costs of producing the pilus or producing phage capsids are incorporated into those biomass equations. We first ran flux variability analysis to look at the amount of flux that we could put through different reactions in the metabolic network and ask, how much overlap was there if we, in the flux that can go through reactions if we're optimizing production of just the host biomass or either of the mobile genetic elements. And we found that about 4% of reactions have non-overlapping fluxes when those different biomass equations are being optimized. So this is 103 reactions which had non-overlapping flux ranges if we were optimizing the host versus the plasmid, and 117 if we're doing the host versus the combined phage and plasmid. And given the composition of hosts in each of these elements, unsurprisingly, we see that most of the increases in flux are associated with nucleotide production and amino acid production, and most of the reduction in flux are associated with lipid production. So our two mobile genetic elements are not requiring lipid, and therefore that drives differences in these allowable flux patterns. We then carried out dynamic flux balance analysis, where now we're looking at either just producing the biomass of the host or putting a lower limit on production of mobile genetic elements and optimizing the host. And with the dynamic flux balance analysis, we're integrating over time. One thing that we saw is that as we account for forcing the cell to produce some of the mobile genetic elements, we see, unsurprisingly, lower growth rate and yield. And what we were more interested in was the implications for the external metabolites. And we did see that in the case of forcing flux or forcing production of the plasmid, of the phage or the plasmid, that we saw more acetate, formate, glycolate, and hexanoate in these infected cells than we did in the wild type. We then made use of a platform called Comets, which allows us to incorporate genome scale metabolic models for multiple species and simulate the growth of those systems. We did this for 3 different community compositions. So E. coli with salmonella, with just methylobacterium, or all 3. And we ran these simulations for the wild type cell, the cell plus the plasmid, or the cell plus the plasmid in the phage. And the simulations suggested that just forcing the cells to produce the building blocks required for making these mobile genetic elements were sufficient to drive pretty dramatic changes in both the total biomass production as well as the composition of all of these different communities, or these 3 different communities. We then went into the lab to test this. And I should have mentioned earlier that all 3 of our strains have unique fluorescent markers, and so we can track the dynamics of all 3 species independently in mixed communities. And what we found was, again, dramatic changes in the composition of these communities as a function of the infection being present. However, pretty poor quantitative match with what we saw in the models. And so we were interested to dig into this deeper. Here, critically, what we're seeing is that across all conditions in which we introduce mobile genetic elements, we're seeing significant increases in the evenness of the community. And so really what we're seeing is that the partners of E. coli increase in frequency dramatically across all of these experimental conditions. So we wanted to dig in and understand that pattern a little bit more. So one question was, are these community effects driven by excretions of the E. coli? So as a starting point for that, we grew monocultures of the E. coli. We then sampled them at mid-log, filtered the supernatant, and did directed metabolomics with LCMS. And we saw what struck us as interesting patterns. We considered that one possibility was that the mobile genetic elements were simply, at some rate, bursting or lysing the cell. So they don't canonically lyse cells, but at some low rate, perhaps they were increasing death rates of the infected cells, and therefore they were leading to or enabling cannibalism. Alternatively, maybe they just make cells more leaky, right? You're jamming a lot of things through a membrane, and so maybe that lets lots of things leak out. But what this pattern suggests to me is, we're seeing relatively few metabolites, which are significantly either more abundant or less abundant in infected cells and uninfected cells. So that suggests to me that it's not just a release of general metabolites, but that it is significant or specific increases in secretion of some metabolites. We were particularly interested in this metabolite, which showed up in both the plasmid infected and the phage infected cells. This metabolite was lactate. We then did HPLC to convince ourselves that this was real, and indeed saw significant increases in the concentration of lactate in the spent media. We then took the spent media from the E. coli monocultures and grew our salmonella and methylobacterium in that spent media, and then again did directed metabolomics. And consistent with the idea that this lactate is influencing the change in species frequencies that we observed, we did see that the lactate was significantly decreased in media that had been depleted by either salmonella or methylobacterium. And finally, we did some experiments where we simply added lactate to our co-cultures and then looked at how that changed the composition. Consistent with the pattern we saw before, our partner species increased in frequency as a result of these additions. However, this increase in frequency was not as dramatic as what we saw from the infected cells. It's interesting that it had the biggest effect in the tripartite community. What we think this means is that lactate is contributing to the changes in the community composition, but certainly isn't the complete story. There are other metabolites that are also, or other factors which are also influencing this change in community composition. We were interested to see this change in lactate specifically because lactate plays a role in in human gut health and in mediating many of the interactions within the gut microbiome. As just one example, the presence of lactate can significantly increase the ability of Salmonella to infect patients. Finally, we wanted to assess the consistency of this effect. And so the first thing we did is we took our plasmids and our phage and threw them into a different E. coli. So in this case, the wild type MG1655, which is able to produce its own methionine. So we changed both the background in which they were introduced, as well as the ecological interaction. So here it's unidirectional facilitation as opposed to obligate mutualism. And we saw, again, this repeated increase in the production or the frequency of the cross-feeding partner. Interestingly, then we tried a different version of the F plasmid, so the POX38, which is significantly smaller. And interestingly, this reduced the effect caused by the plasmid itself. We still see a significant effect of the phage in the presence of this plasmid, but the plasmid itself lowers that cost. So, in quick conclusions of what I think are some interesting patterns, we showed that MGE can generate metabolic conflict with the host using the genome scale metabolic models. We showed that MGE can alter community composition using both the models. We showed that just the metabolic costs of the building blocks are sufficient to drive changes in community composition. We also showed that experimentally. We showed that MGE caused E. coli to secrete lactate and that there's a consistent effect across host, but not plasmid. In the spirit of highlighting some of the caveats of the work, which Wenying asked us to do, so I started by saying mobile genetic elements are really prevalent. And so I think the ability of mobile genetic elements to drive changes in community composition has potential important implications. We chose mobile genetic elements which were specifically costly, producing many proteins, so producing all of those phage capsids or producing a pilus. Certainly, I wouldn't argue that all mobile genetic elements are going to be nearly this costly. So there will be many, for example, prophage or many plasmids which aren't producing pili, which will have much lower cost than this. But there are still many examples from other systems, such as the Lopakhin lab and some of the great work from the Duhaime lab, showing that there are still frequently costs. I think what our work suggests is that these costs are not purely metabolic and may often be driven by changes in gene expression, for example. So with that, I will let people rest. This was all the work or primarily the work of Abe Bessesi, who was a grad student in my lab and is now off to do a great postdoc at NYU. We had some great input from collaborators, and I'd be happy to take any questions. Thanks a lot, y'all.
That was really interesting to think about mobile elements as like a way of engineering a community, like changing its function.
Yep.
And I guess one of the challenges with like how do you predict the effect in a given community? And I wonder if that's like how, I don't know if you had any thoughts about like whether that you think that's feasible or how that might be done.
Yeah. Boy, I sure hope so. So I was really hoping that the genome scale metabolic models would give us a place to start. And they certainly gave us a place to start and a null model to compare against, which I think was useful. I think that I don't have great answers yet on how we could predict these exact effects. I don't understand what's driving the effect in this system yet. So I'm mostly presenting you with a pattern without great theory or great biological answers. And maybe people in this room will help us generate some of those answers. I guess what I would say is that I do think that there will be metabolic components, which once we have a better understanding of the rules, will be generalizable. It clearly won't be perfectly predictive, but I think there are generalities that exist. And so that's sort of a direction we're excited about going, is trying to, this was a small subset of things, right? And so we're excited to test many more genetic elements across many more strains, and start to pull out some of those general patterns.
Huge data set, and then of course data-driven approaches might be useful in that case.
And if you have some data that might work for that, I'd love to talk more. Yep. Yeah.
Sorry, I was wondering about, in some phages, especially cyanobacterial phages, people find metabolic gene being encoded by phage.
Yeah. Yep.
Did you have anything like that in this system? Do you know the phage genome?
Yes. Yeah. So that's a great point, and that's one of the points I was trying to make early on, is that I think that's generally the way that people think about mobile genetic elements influencing the metabolism of cells, is through providing novel metabolic enzymes. In this case, there were not novel metabolic enzymes. So it was purely just the rewiring that is the result of taking over the cell, that we were focused on here. So yeah, so that we conscientiously tried to avoid that in this case. Yeah.
Really beautiful stuff. So could you speculate a little bit why it's lactate that E. coli tends to spit out? Does that anticipated at all from your FBA, even if the FBA doesn't anticipate the absolute biomass trends?
Yeah. So FBA did suggest that there would be organic acids. Lactate is an organic acid. However, the model doesn't tend to suggest lactate specifically. One of the hypotheses that I was intrigued by going into this was that maybe what was happening is that because you have the F. pilus forming in the plasma membrane, and then also the filamentous phage sort of pushing through that membrane, that maybe what you're doing is you're competing for real estate in the membrane with electron transport chains. And so that's forcing the cell to go from respiration to fermentation, and that's driving the increase in organic acid. We don't have any, none of our data directly supports that. And in fact, the fact that when we had the smaller F plasmid, which is still producing pili, we didn't see strong effects of lactate production, I think argues against that. So that's all to say, no, I don't know. I think there's some, that's a sort of a continued direction we're wanting to go. I guess another thing that I will say is that, the genome scale metabolic models are basically evolutionary optimality models, and this clearly we just introduced a perturbation, and so it's not surprising that it's not optimal. So another direction that I'm excited about for us to go is to start evolving these and say, as you start, getting these to get closer to an evolutionary optimum, do they then start mirroring some of the things that we see in the models a little bit more? So I don't have an answer for your question, but
one question at the back there.
Just quick question: Did the pH change during growth for all these different combinations?
Yeah, we did not see any changes in the pH. So we're using M9, which is pretty highly buffered, and so I wouldn't expect dramatic changes in the pH. But pH changes could certainly be one thing that is influencing the effects of these. But we didn't see evidence for that in our experience.
Just to follow up on this lactate, your hypothesis for lactate, did you see any other end products of mixed acid fermentation like formate or ethanol?
We did not. So it was, the lactate, the reason that we sort of cued in on the lactate was that it was the primary organic acid that we observed, so the primary resource that we could see being consumable.
That's very interesting.
Yeah. You would have predicted.
Yeah, well, if the idea with the membrane space real estate for respiration is true, they could switch to mixed acid fermentation.
Sure. Yeah.
And are done with oxygen business. But then you would expect some ethanol and formate.
Yeah. Right. Yep. Yeah. So that's right. Yeah. And we didn't see evidence of that here.
All right. Maybe one last question, and then we'll wrap it up.
I actually want to follow up on that question. FBA models typically will not excrete anything unless you put in some kind of a explicit constraint, either on protein fractions or the membrane constraints. So did you put some kind of overflow metabolism constraints in your models so as to predict?
So there were some constraints on the oxygen uptake, which I think led to some of that release. And then I think actually what's happening is they're depleting the oxygen so they can't retake it up. And so even on our wild type, we see some initial release of it, but then it's reconsumed. And so it's just in these models where they can't take it back up. But some of the approaches on molecular crowding or expression constraints would be another mechanism that could explain some of this. We tried some of those and didn't find strong support for at least driving the lactate production. But I think there are more things to try there as well. So, thank you.